Hi everybody, and welcome to our module on innate immunity. The easiest way to understand the immune system at a basic level is to think of it as a series of barriers to infection. So let's imagine, for example, that we have a bacteria here, or maybe a helminth like I've drawn here, or maybe a virus like I've drawn here. And these pathogens want to get inside the body because inside the body they can grow and thrive. Well, the first barrier that keeps them from getting inside the body is the skin and the mucous membranes. We often don't think of this as part of the immune system, but the reality is that there are many pathogens we're exposed to every day that never gain entry to the body because they can't get past these basic layers. But once a pathogen gets past the skin or the mucous membranes, it encounters what most of us think of as the immune system. And the first thing it encounters when it gets past the skin and mucous membranes is the innate immune system. This is a system made up of cells like macrophages and neutrophils and proteins like complement, and they attack and try to kill these pathogens as soon as they get in the body. As we will learn, however, in this module and other modules, the innate immune system is not so good at dealing with overwhelming infections or dealing with viruses. So once a pathogen gets beyond the ability of the innate immune system to control the infection, it then will encounter the third barrier, and that is the adaptive immune system. The adaptive immune system is made up of B cells and T cells. These cells create an immune response that is highly specific to an invader. They devise systems to kill one specific invader. It takes time for them to mount their response, but once they do so, this is the most powerful portion of the immune system. Another feature of the adaptive immune system that's unique is that it has memory. So if you get infected by the same pathogen 2 times, the second infection will elicit a quicker and more powerful response from the adaptive immune system. So on this slide, I've summarized some of the key differences between the innate and the adaptive immune systems. The innate immune system is a fast-acting system. It is standing by, ready to attack any pathogen immediately if it gets beyond the skin and the mucous membranes. In contrast, the adaptive system is slow. It takes a few days to mount its response. But as I said before, once it does, it's a much more powerful response than what you get from the innate immune system. The innate immune system is a non-specific reaction. The same cells respond in the same way to many different invaders. The adaptive immune system is a highly specific response. As we will see in other modules, B and T cells proliferate in response to unique antigens on unique cells to try and repel and kill that invader. And then finally, the innate immune system has no memory, so a second infection elicits the same response as the first infection. The adaptive immune system has that important feature called memory, where the second infection elicits a faster and more robust response than the first. Before we can talk about the innate immune system, we have to define a number of terms that are very important for discussing immunology. And the first term is that of antigen presentation. So I want you to imagine that we have some tissue in the body here, and it has been infected by a bacterial cell. I'll draw a little circle around a B here for the bacterial cell. You should know that the innate immune system can react directly to antigens present on the bacterial cell. So for example, as we will see, macrophages can eat these bacteria. It's a process called phagocytosis, and they can do that directly just by sensing the presence of the bacteria. On the other hand, cells of the adaptive immune system, the most important of which are the T cells, cannot react directly to this bacteria. The bacteria can be right next to the T cell, and the T cell won't do anything at all. The only way the T cell can react to this bacteria is if the bacteria is first consumed by a cell called an antigen-presenting cell, or an APC. That antigen-presenting cell will then gobble up the bacteria and present little pieces of the bacterial peptides on its surface, and then the T cell can see the bacterial pathogen and react to it. So T cells in the adaptive immune system require antigen presentation in order to activate. And the reason this system evolved is because the T cell response is incredibly powerful. And if T cells could react to anything out in nature, they would probably react too much and we would die from autoimmune diseases. By requiring antigens to be presented to T cells by antigen-presenting cells, it fine-tunes the system so that the adaptive immune system only responds to important invaders. So I've summarized these points for you on this slide here. The innate system can be activated by free antigen, like that bacterial cell floating out in the tissue. Pathogenic molecules can be detected freely in the blood and tissue. On the other hand, the adaptive immune system, especially the T cells, require antigen presentation. Pathogens must be engulfed by cells and broken down. Pieces of the proteins have to be transported to the surface, and the antigen has to be presented to the T cells for activation. Another important concept for discussing immunology is the concept of cytokines. These are cell signaling proteins that are often released by immune cells, and they travel locally to stimulate an inflammatory response. And there are a number of subsets of cytokines, and you have to be familiar with these terms. So chemotaxis is the process of attracting immune system cells. So cytokines that attract immune system cells are called chemokines. Interleukins are another subset of cytokines. They were given this name because these proteins travel between leukocytes, and they're named with numbers like IL-1 and IL-2. Some cytokines are called tumor necrosis factors, and these were named because they can cause tumor cell death, but they also are used by white blood cells to trigger immune responses. And then other cytokines are called transforming growth factors and also interferons. The interferons got their name because they interfere with viral replication. And then the final concept we need to define before discussing the innate immune system is that of the CD structures, or the cluster of differentiation structures on cells. So CD structures are cellular surface molecules, and they have names like CD3 and CD4 and CD8, and they are found on many immune cells, especially T cells and B cells. And we use them to identify cell types. So for example, when we say we have a CD8 T cell, we're talking about a specific subtype of T cell, and it's defined by the presence of this CD8 molecule on its surface. Also, some of the CD molecules are used as receptors and cell binding sites, and we'll talk about those in the remainder of this module. So let's start our discussion of the innate immune system by listing the key players in this system. And the first set of key players are the phagocytes. These are cells that essentially eat pathogens that get inside the body. The first group are the macrophages. This is the hallmark cell of innate immunity, as we shall see. And then the second group are the neutrophils, which are cells that are essentially on call, and they can be called in to assist the macrophages when an infection is taking place. The next key element of innate immunity is the complement system. This is a series of proteins that can be activated inside the body to kill pathogens. And then finally, there are natural killer cells, eosinophils, and mast cells, and basophils. And in the remainder of this module, we're going to talk about all of these elements of innate immunity. So first, let's talk about how those elements of the innate immune system recognize pathogens that have gotten past the skin and mucous membranes. And you should know that all those elements of the innate immune system can recognize molecules that are foreign. And it does this by looking for structures on pathogens called pathogen-associated molecular patterns. These are patterns of molecules that are present on many microbes, but not usually present on human cells. And the way the innate immune system recognizes these structures is by the use of pattern recognition receptors. And one of the most important classes of pattern recognition receptors are the toll-like receptors, or TLRs. These are found on cells like macrophages and dendritic cells and mast cells, and we'll talk about this in a minute. And once these cells recognize the pattern-associated molecular patterns, or the PAMPs, they then secrete cytokines which trigger an inflammatory response. So what are some of the important pattern-associated molecular patterns that the innate immune system responds to? One of the most important ones is endotoxin, or lipopolysaccharides, which are found in gram-negative bacteria. Lipopolysaccharides can bind to lipopolysaccharide binding protein, which is found in plasma, and then that complex can bind CD14 on macrophages, and I've written this in bold because it's high yield to remember that CD14 binds lipopolysaccharide. Once CD14 is bound, it will trigger a toll-like receptor called TLR4, and this will lead to a sequence of events which leads to production of cytokines by macrophages, including IL-1, IL-6, IL-8, and tumor necrosis factor. We'll talk about all these cytokines in a few slides. The innate immune system can also recognize peptidoglycan in the cell wall of bacteria. If bacteria get inside a macrophage, for example, there are receptors called NOD receptors that are intracellular. NOD stands for nucleotide binding oligomerization domain, and that's a mouthful. But once peptidoglycan binds these NOD receptors, this leads to cytokine production. Other patterns that are recognized by the innate immune system include mannose. Mannose is a polysaccharide found on bacteria and yeast. And there is a protein in the body called mannose binding lectin that comes from the liver. And once it binds to mannose, this can activate the lectin pathway of the complement system, which we'll talk about in another module. Another pattern recognized by the innate immune system is lipoteichoic acid. These are proteins found on gram-positive bacteria. Also, double-stranded RNA. Double-stranded RNA is never found in normal human cells, but many viruses are RNA viruses. Some of them carry double-stranded RNA in their genome. Others have a single strand of RNA, but they will briefly become double-stranded when they are copying that RNA genome. So the innate immune system is programmed to recognize double-stranded RNA. Another pattern that the innate immune system recognizes is unmethylated DNA. So most of the DNA in human cells is methylated when the innate immune system sees lots of unmethylated DNA that is more commonly found in bacterial cells, and it will react to that.